I'd like to request all the speakers to occupy their respective places on the stage. A very good morning to all. I'm Naresh, currently working as a assistant professor at the PhD Department of Chemistry, Shantadas College, and it's my absolute pleasure to extend a hearty welcome to each one of you present here at the International Seminar on Neurodegenerative Disorders and Inflammation, organized by the Department of Botany, Microbiology, and IUSC, Shantadas College. The seminar aims to explore the dynamics of neurodegenerative diseases such as Alzheimer's, Parkinson's, and Parkinson's that has posed significant challenges to the healthcare system worldwide due to their progressive nature and lack of therapeutic treatments. It also aims to explore the role of inflammatory resources and their regulation towards epidemic mechanisms that are pivotal in the onset and progression of these diseases. We are thrilled to witness such enthusiastic participants and audience gathered to deepen their understanding in this specific topic. Before we commence our event, it's time to seek blessings, wisdom, and guidance through the prayer. Now I would like to call upon Sagar S., first year BSc, to lead us into the prayer. கடலுக்காய் கட்டி இருகண்களமாய் கட்டி வந்தாகிசினார் கைவதனபியும் ಶರದಿ ಚರಣವನೆತ್ತಿ ನಾಟ್ಯವೇ ಪ್ರಣವ ಸಂಗೀತ ಗಣಪತಿಯೇ ನಮಃ ಪ್ರಣವ ಸಂಗೀತ ಗಣಪತಿಯೇ ನಮಃ പൂജകൾ पाशाकुशल श्रीगणनाथ श्रीशाशित पलकामितनाथ पाशाकुशल श्रीगणनाथ श्रीशाशित पलकामितनाथ वसवसन्नुत शंकर पुत्रा शंकर पुत्रा मुझे गुरु मुझे गुरु सुरमुनि पूजित सुंदर चरणा सुरमुनि पूजित सुंदर चरणा दुरित कड़ल कड़गुण पूर्णा सुरमुनि पूजित सुंदर चरणा दुरित कड़ल कड़गुण पूर्णा गिरिजानंदन मंगल विसरै गिरिजानंदन मंगल विसरै स्मरि शिवकार्य कैगूडि पुरे स्मरि शिवकार्य कैगूडि पुरे पूज्य विनायक मूजगपुरे विनायक पूज्य Thank you, Father, for the soulful prayer. Hope it has instilled a sense of calmness in all of us. Now, to formally commence this inaugural session, I would like to invite Srimati Sugandhi S.A., Associate Professor, Department of Physics, Sharda College, to welcome the distinguished speakers. Over to you, ma'am.
Good morning, everyone. On behalf of Sharda College, Mysuru, I welcome you all to the international seminar, Neurodegenerative Disorders and Information. We are honored to have such a distinguished group of dignitaries for today's international seminar. Let's open our minds and hearts to new ideas as we come together to learn new facts and shared knowledge. On behalf of this prestigious institution, I wholeheartedly welcome our Honorable Vice Chancellor, Professor Lokanath, University of Mysore, to this international seminar. Welcome you, sir. It also an honor for us to welcome Professor K. Sandeep Prabhu, Head of Immunology and Molecular Toxicology, the Pennsylvania State University, United States of America. Welcome, sir. It gives me immense pleasure to welcome our Honorary Secretary, Shri Chandrasekhar Sir, Shardavlas Educational Institute. Welcome, sir. Now is the time to welcome Dr. T.K. Umesh, Senior County Counsel and former Vice Chancellor, University of Mysore. Welcome, you, sir. I also welcome our dear Principal, Dr. N. Devika, for this international seminar. I welcome you, madam. I also welcome all the honorary advisory committee members to this international seminar and Dr. D.S. Baswaka, Chairperson, Department of Studies of Zoology, University of Mysore. Welcome, you, sir. A warm welcome to all GC members, all participants, few retired staffs, electronic media, colleagues, students, and technical team. Thank you for your support. I welcome you all and welcome all the August gathering sitting over here. Thank you one and all.
Thank you, ma'am. I would also like to welcome you to this event. Just as right when you start this, knowledge events. And so complementing this, she launched our events by lighting the lamp. I would like to call upon the dignitaries on the desk to light the lamp. We have with us today Professor A.K. Loknath sir, who is the Honorable Vice Chancellor of the University of Mysore. Professor A.K. Loknath sir has received his doctorate degree in physics from the University of Mysore, and he has index of around 30 and over more than 3,900 citations. His research interests are crystallography and structural biology. Professor has been published more than 400 research articles in reputed and highly impacted international journals, and he has delivered over 47 invited lectures at various international and national conferences, both India and abroad. Professor has more than 1,000 crystal structural data that has been deposited in CCDC and over 70 protein structure that has been deposited in protein data bank. Further, Professor Loknath sir was also awarded with prestigious awards. Some of the prominent ones are Sir C.V. Raman Young Scientist Award from the Government of Karnataka, Research Faculty Award by Career 360 New Delhi, and he has also granted with various prestigious research projects from prestigious governing bodies like ESC, UGC, UPE, and so on. Over 11 PhD students have been awarded doctoral degree, and currently 9 research scholars are working under his guidance. He is also the member of various global scientific societies like American Crystallographic Association, the International Union of Crystallography, Japanese Biochemical Society, Korean Crystallographic Association, and so on. With this brief introduction, I would like to call upon Professor A.K. Loknath sir to address the utmost gathering. Over to you, sir.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ವೆರಿ ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಟು ಎವ್ರಿವನ್. K. Sundari Department Head and Professor of Immunology and Molecular Toxicology, Department of Veterinary and Biomedical Sciences, Pennsylvania State University, USA. He is Chandrasekhar, Honorary Secretary, Andhra Transportation Institute. Mr. T. Umesh, my teacher, my mentor, my guru, so many things. So and now he is as a professor, the chairperson of the department, vice chancellor of the university. Now he is here as a senior governing council member. Dr. Devika, principal, Chandra College, and all the participants, particularly faculty from different colleges, universities, and teaching and non-teaching staff of this college, the students, and media persons. I'm extremely very happy that to inaugurate this international seminar on neurodegenerative disorders and inflammation. I came to the Scholar College many times, many days, many capacities. This is the first time I'm coming to here to inaugurate this national seminar. That too, particularly to the interest of my knowledge, the subject is very interesting. So this subject itself, so that this is a multidisciplinary subject where people are students are eager to learn interdisciplinary subjects. So that today, Dr. Sandeep Prabhu will definitely shed light on these issues, which is very relevant nowadays. I'm just addressing to the students. Anyway, to speak about this scientifically, I am addressing you. There are 3 things you notice in higher education. You are all in higher education, means you are in graduation. Higher education is the one where you can create the world. This is the where your life starts. Those who are entering higher education means they will be going to higher education. So all the people on the stage and on this Pindaya, they are all recognized just because of higher education. Therefore, higher education will change life. So students, be serious about your education. That too particular higher education, it will give you a lot of things. For this, so you know that in India the number of students studying higher education is very, very large. Probably you do not know there are around 30 million students, as per knowledge, 30 million students are studying higher education. So So that is much more than the population of many countries. So 30 million students are looking at this competitive world. So you need to be very careful. You need to be learn many things at a time. You should have to do something else. Therefore, the competition is very competitive. And the second thing is, this is all possible to learn, to study well, to do some good things, because you should learn the skill sets. Now, just having a degree, just having an education is not enough. You need to learn the skills. So, as you know that today we are all living in modern age where we use the technology and information. And talking about technology, you can take every student will have a mobile, and every student will have an internet, right? And you use WhatsApp regularly. Only you can send the message and receive the messages, as I know, and forward the message. But you do not create any message. I am urging you, you should create a good message. Then you will have some idea, some thought. Therefore, you have a technology like mobile, internet. In the internet, you have a information, right? Technology and information together will lead to a lot of things. Therefore, you need to learn what best you can do with this technology. Thereby, they can learn the knowledge. Education is not just seeking a job or getting a degree. Education is to get knowledge. Once you get a knowledge, you can be able to learn whatever you want to do. That's what I'm saying. Probably you can ask Sandeep Prabhu or your Professor Gunesh. They studied something, after that they do something else. So that means the basic education is very important. Therefore, the fifth thing, education means knowledge. So you have to gain knowledge. For this particular student, you should listen the lecture. Probably you might not understand the interest or what are the objectives of this research. This will give you some idea how can you understand the new things. So we will try the possibilities, to explore the possibilities, what way they can do with this education. So therefore, we should be very careful. He is talking about problems that disorder. He is talking about some disorder disease. I remember. So these are very important. Say for a human, any human will have some disease. These diseases will be studied in different ways. So that by taking biochemistry, or maybe something like a computation studies. So whatever the technology uses, so you have a big data, data science. Once you have the data, then you can model the things such a way that you can be able to explore the possibilities of solution to these problems. So therefore, all the students should take it serious to learn this and to study well. So there is no shortcut route for study as the great section of every student should have a vision. What is the vision? Vision is the goal. So if I am completing my BSc in 21 years, if I complete MSc in 23 years, what will be the next? You should not have a question, you should have a plan. So then you can achieve the goal. So therefore, you try to do well. Always you have to work hard. Sincerity is more important. So there are two things to do there in India. One is the quality, another is the quantity. We are number is very big, we have a quantity. So what about the quality? Quality is there, but the quality comes down. So we need to impress on the quality, therefore, to press on this and learn and explore the possibilities of new things, what is happening in and around you, and so we have to use this technology to learn these things. So and be alert to the modern science. So you can see that every day people are talking about artificial intelligence. Just I'm talking to Professor Prabhu. So artificial intelligence is very essential, but only if you have intelligence, right? So you have to think about this. So that's what I'm telling. Even if you use internet, even if you use technology, which one interests you, that you have to think about it. So with that, so I'm very happy that this Southern College is arranging this famous international seminar. Therefore, Professor Prabhu is doing all the time university to enlighten you, to know about the new methods of disorders and diseases. So certainly this lecture will be very interesting. So you will learn the things and you have to think that what are the techniques that they are used to learn these diseases. Those things you have to understand. So with these words, I am very thankful to you, sir, for giving me the opportunity to come over here and to inaugurate this important national seminar. So once again, thank you to all. Thank you.
Thank you for your wonderful words of encouragement, sir. As it is a custom at Chandala's College to honor our guest, I request all the dignitaries on the dais to felicitate our chief guest of today's inaugural session, Professor A.K. Loknath, sir. Thank you all the dignitaries for honoring Professor A.K. Loknath, sir. Without further ado, I take great pride and honor in inviting Dr. Nirvana, Principal Chandala's College, Mysore, for the presidential address. Let's welcome with a round of applause and the stage is all yours.
A very good morning to one and all present here. Esteemed Chief Guest, Professor Loknath Reddy, Honorable Vice Chancellor, University of Mysore, Eloquent Speaker, Professor Sandeep Prabhu, Department, Head and Professor of Epidemiology and Toxicology, Pennsylvania State University, United States of America. Distinguished guests of honor, Mr. M. Chandrasekhar, Honorary Secretary, Chandala's Education Institutions, and Professor J.K. Umesh, former Vice Chancellor and Senior University Member, Chandala's Education Institutions, faculties, researchers, and students gathered here from various institutions. It's a privilege to stand before you today at this introduction seminar on neurodegenerative disorders and information. As we gather here from various corners, our shared commitment in combating these devastating health conditions has united us with a common purpose. Neurodegenerative diseases such as Alzheimer's, Parkinson's, and so on represent some of the most formidable challenges facing modern medicine. The social impact of these diseases on individuals and families is really immense, both in terms of suffering and also in terms of economy. Similarly, inflammation, whether as a cause or a consequence, plays a significant role in the progression of these diseases, adding complexity to our understanding and treatment approaches. As we delve deeper into the intricacies of neurodegenerative disorders and inflammations, new questions and challenges emerge for which we can find answers from scientific sections like this. We believe that by fostering an ecosystem of innovation and exchange of information, forums like this seminar will serve as catalysts for advancing scientific knowledge and translating it into tangible benefits for mankind. Therefore, our Chandala's education institutions, in specific Chandala's College, has organized this seminar thinking that the collective efforts of researchers and clinicians around the world would yield remarkable insights into the underlying mechanisms of neurodegeneration and inflammation. As stewards of knowledge, we, the higher education institutions, have a moral imperative to address these issues and also to see that this type of forums help in finding solutions. and we are committed to do so by bringing together experts from around the world to share their insights and explore innovative solutions to these challenging health conditions. In closing, let us recommit ourselves to the noble cause of preserving human dignity. Hope by harnessing the power of collaboration and innovation, we can overcome the formidable challenges posed by neurodegenerative disorders and inflammation and pave the way for a healthier, more resilient future for all. Accomplished researchers and doctors who are delivering talks today will be giving lots of insights into the subject of interest and benefit the participants. I appreciate all those who are participating in this seminar for their commitment towards the academic growth and pursuit of knowledge. I wish every participant will be benefited in this regard. I also wish a comfortable stay for everyone in our college premises. Thank you, one and all.
Dr. Devika, for sharing your valuable insights and setting a powerful and wonderful vision for the future of our institution. Now let me call upon Mr. Yashwant, Assistant Professor, Department of Botany, to deliver the vote of thanks.
Respected dignitaries, luminaries, respected delegates, faculties, and research scholars of various institutions, students, my colleagues, a very good morning to all present here. I feel greatly honored and privileged to be a part of this international seminar on neurodegenerative disorders and inflammation, organized by the Departments of Botany and Microbiology in association with IPAC, Sharda College, Mysore. Gratitude sweetens the smallest moments. It gives me immense pleasure to propose vote of thanks for this inaugural session of International Seminar on Neurodegenerative Disorders and Inflammation. I take this opportunity to acknowledge everyone who has witnessed this auspicious movement. To start with, I extend a deep sense of gratitude to Professor N.K. Loknath, Honorable Vice Chancellor, University of Mysore, Mysore, who, in spite of his busy schedule, accepted our invitation and graced this occasion by inaugurating this event. On behalf of Chandras College and on my personal behalf, I thank you, sir, for your esteemed presence. Thank you. I'm also thankful to Professor K. Sandeep Prabhu, Department Head and Professor of Immunology and Molecular Toxicology, the Pennsylvania State University, USA, for witnessing this inaugural session of today's event. Thank you, sir. My sincere thanks to Sri N. Chandrasekhara, Honorable Secretary, Sharda Educational Institutions, Mysuru, for his constant support and guidance for organizing such kind of events in the college. Thank you, sir. I would also express my gratitude to Professor P.K. Umesh, former Vice Chancellor, University of Mysore, and one of governing council members of the college for his appreciated presence to this international seminar. Thank you, sir. I express my sincere gratitude to Dr. Devika M., Principal, Sharda College, who has been the supporting pillar of this event and who played a major role in organizing this international seminar. Thank you, ma'am. I express thanks from the bottom of my heart to all the faculties, research scholars, students of various institutions, and also the faculties and students of Chandra College who has witnessed this inaugural session. Thank you all. I would not forget to acknowledge non-teaching staff of the college and media partners who has come to this inaugural session. Thank you one and all.
It is a privilege to introduce the fourth person to the August gathering, our first keynote speaker, Professor K. Sandeep Prabhu, who is a renowned expert in the field of inflammation and immunology, who has an experience of 42 and ideal index of 67, and more than 4,700 citations. His research and teaching interests are microbial biology, hematology, analysis, targeted leukemia stem cells, nutritional biology, pharmacology, and immunotoxicology. Professor Prabhu has accomplished master's doctorate degrees from the University of Mysore, pursued postdoctoral research at Pennsylvania State University. He was awarded gold medal during his master's. He started his career from UPR Mysore as a junior research fellow at the Department of Biochemistry. Presently, Professor Prabhu is the head and Professor of Immunology and Molecular Toxicology, Department of Veterinary and Biomedical Sciences at the Pennsylvania State University, United States of America. Professor has a vast teaching experience of more than 20 years and research experience of more than 30 years. Professor Prabhu has been honored with various prestigious awards. He was awarded CSIR Government of India Travel Award for traveling in the 8th International Psychology Symposium at Hungary. He was awarded the 8th Investigator Award from Mexico Research Foundation in Canada. He has received the Innovator Award from Pennsylvania State Research Foundation. Professor has bestowed the foreign faculty travel award by Global Initiative of Academic Networks. He has been the invited speaker for many important occasions and international conferences in countries like Hungary, Puerto Rico, France, China, Mexico, and all around the states of India. Presently, Dr. Prabhu is the ad hoc reviewer for the Biochemistry Journal and the Journal of Biochemical Chemistry, the Journal of Physical Chemistry. He is also the review editor at Frontiers in Nutrition and also the editorial board member of the Nutrition Journal. The professor has authored more than 160 research articles, published several book chapters, and has 30 patents under his name. We look forward for Professor Sing's presentation on resolution of inflammation and new therapeutic cardio. Ladies and gentlemen, please welcome Professor K. Sandeep Prabhu.
Okay, you can all hear me well? Okay. Once again, namaskar everyone. Thank you, Professor Devika, for the invitation, and thanks to all of you for coming here. It's a privilege and an honor to be here at Shadavilas Institute, and I hope to tell you something new about a new therapeutic market of treating inflammatory diseases, including neuroinflammatory diseases. I just want to give you a quick overview of my university. This is the Pennsylvania State University. We are in central Pennsylvania. This is the map of the United States. And you can see here, this is where the state of Pennsylvania is. And we are about 20 campuses with about 90,000 students total. And in our campus there, shown in red star, we have about 45,000 students with a big annual total budget of $8.6 billion. Our president is of Indian origin, of Neurodegenerative, and so you can see India has made a big mark even in the education sector in the US. So the university started as a farmers' high school in 1855, and that building got transformed to this building currently. Okay, it started as a farmers' high school and then became a state university. It's a state-funded university. So agriculture, health, human health, animal health, environmental health, all of those are of great interest to all of us. My lab in particular is looking at how we treat inflammation that is caused by environmental toxicants, pathogens, infectious disease, pathogens or nutrition deficiencies by working on what is called optimal nutrition to help suppress inflammation by increasing resolution of inflammation. So in my lab, I am interested in two particular nutrients. One is for the omega-3 polyunsaturated fatty acids, aka fish oil type fatty acids. And then other one is a micronutrient for selenium. Okay, you all know inflammation is important, but I don't know if you know inflammation is also labeled as the secret killer. Because if you look at myocardial disease or cardiac diseases, you look at cancer, or you look at neurodegenerative diseases like Alzheimer's, you can see inflammation is very important. If you can treat inflammation, you can actually treat many pathologies that lead to either a cause or it's a consequence, making inflammation treatable or those diseases treatable. So my dad is interested in understanding these paradigms or various paradigms that are there and trying to see how can we come up with new therapeutics. As you know, these are the five cardinal signs of inflammation. That is heat, redness, swelling, pain, loss of function. This is, I didn't make this. This is from, Greek philosophers long, long ago who saw this, that inflammation can be seen in all of them. And that can lead to many diseases. Uh, so before we try to understand inflammation, I want to tell you something about what actually causes inflammation. Yes, your pathogens, your viruses, your all heard of coronavirus, you've heard of bacterial infections, you've heard of influenza infections. All these infections actually target some of your cells. And when you target cells in your immune system that has two arms, one is called the innate immune system, the other one is adaptive immune system. The innate system is like your first responders, like your police or fire brigade, all these people. Those are the innate arm of the immune system, and the adaptive arm comes later. It can come and start in waves, and that actually helps to give you long-term protection. So what gives you protection early on are those innate immune cells. But sometimes what happens is the innate immune cells can also go crazy, and they can lose their control, and that's when inflammation goes uncontrolled. Okay? And within the innate system, we have many different cell types. That is, seen in the form of epithelial cells, dendritic cells, mast cells, and so on. Of those, innate cells, there is one particular cell type that is of great interest to a lot of people, including me. Those are called macrophages. How many of you have heard of macrophages? Okay, very few. Macrophages are pretty much called professional phagocytes. What they do is, when they see something, they go and take it and eat it. Okay, it can be bacteria, it can be, lot of junk matter. They can eat. But they are not just phagocytes. They do a lot of things because these macrophages arise from what is called the yolk cell. Particularly those in the zoology field know more about the yolk cell, right? And from the yolk cell, these macrophage lineages actually come to the fetal liver. From the fetal liver, it goes to the bone marrow. In the bone marrow, there are a lot of stem cells that lead to the formation of these macrophages. Now, those macrophages then go in an adult. They will go to every organ in the body and will populate those cells or those organs. For example, since the conference is about neurodegeneration, the most important organ in your body is the brain, right? So these macrophages go there and set up their shop. That's where they're called microglial cells. Then they help in neuronal patterning, they help in fluid balance, they help in reducing inflammation, they help in a lot of things. But these macrophages, when they get out of control, they can actually help in neurodegeneration. So you can see macrophages have both sides. They are good, but they are also bad. So somehow you have to keep the macrophages clean. You have to keep them controlled. Okay? And if you look at the heart, there are macrophages. Anyone who has a heart attack, within about a day of having a heart attack, macrophages are mobilized from your femur. They come to the heart and then try to repair the heart. So we have tracked those macrophages coming into the heart when somebody has a heart attack, which means they are trying to help in restoring. In the liver, there are some cells called Kupffer cells. They are also macrophages. So macrophages are present pretty much everywhere in every organ you can think of. And in the lungs, you have lung macrophages, that's what alveolar macrophages. But what is very important to this talk is this microglial cell, which is part of the central nervous system, that actually helps in eliminating old and dead neurons, and it helps control the immunity in the brain. So, one of the very interesting things about these macrophages are they are extremely plastic. What I mean is they can easily bend, or they can change their phenotypes. Okay? So they are somewhat, to relate to, they are like another me. Okay? You will talk this way or you can talk this way depending on the cytokines. Alright? I don't know if Nadan really talked about cytokines, but at least I'm giving you an example. So what starts is with this naive macrophage. When it is inflamed, it becomes a highly pro-inflammatory macrophage. But when there are anti-inflammatory cytokines, then it immediately switches to an anti-inflammatory macrophage. So macrophages can go back and forth. Something very similar to T cells. T cells can also do this. So the immune system is so well organized that it can switch according to the scenarios that we often encounter. So if you look at Alzheimer's, for example, inflammation in Alzheimer's is caused by, at least from what we know, is caused by the production of these amyloid beta proteins. Now, amyloid beta proteins come from this amyloid precursor protein called APP. I'm sorry, this pointer doesn't work on the screen. But what you get is the amyloid beta peptides. Now, this amyloid beta peptides can actually, form these big aggregates, and those aggregates lead to the formation of what are called tau tangles. These tau tangles can interact with your neurons and cause the neurons to be stressed. But at the same time, these tau tangles and some of these peptides, amyloid peptides, can be cleared by your microglia. However, this does not happen all the time. The macrophages do not just clear them, but instead they can get activated and increase inflammation. So, what is important is you need to keep the macrophages under control. Okay? So, this is in Alzheimer's. Okay, so if you next see in Parkinson's 2, go back on the full hand. Yeah, go back. Go back. Back. If you look at amyotrophic lateral sclerosis, this is also a neurodegenerative disease, even there you see that microglia are important. Okay. So, so in neurodegeneration, what is very important is inflammation that is caused by increased activation of some of the transcription factors such as SNAP, NF-κB, and so on, lead to reactive oxygen species and a lot of cytokines, and all that helps in inflaming the system. So if you know what is actually dysregulated, you can control that and you can treat the neuroinflammation. So the questions are, what are SPMs and how do you integrate NF-κB? How many of you have heard of NF-κB? Anyone? Have you heard of transcription factors? Okay. That is a transcription factor. That transcription factor is extremely sensitive to stress. If there is stress in the system, it will get activated and increase the stress even more. Okay? So we need to somehow target NF-κB. So that actually leads us to what is called primary resolution of inflammation, where if you can resolve inflammation or you can suppress NF-κB and others, you can actually help treat some of these neurodegenerative diseases. So the thing is, it's not very easy because inflammation and primary resolution is not just by macrophages. It's a team activity. It is like a cricket team, where you have to have multiple different cell types where there is inflammation that goes up, then comes the resolution part and it resolves. However, in people who have neurodegenerative diseases, this curve is like this. So there is no resolution at all, it's always inflamed. So somehow you have to bend this curve and do it this way. Okay, that is the challenge. And for that to happen, you have to have many cell types, like your microglia, like monocytes, macrophages, or you need to make sure it's from neutrophils, you have neurons. All these need to work in tandem to actually make this happen. Okay? Okay, so my lab is interested, as I mentioned earlier, it is interested in a nutrient called selenium. How many of you have heard of selenium? Okay. So selenium is a metal, so an element in the oxygen sulfur family. Okay, it's right here. And this is a element that was discovered about 200 plus years ago by a Swedish biochemist. It's an essential trace element. You don't need much, but you need a lot of it. And deficiency of selenium is known to be seen or is known to cause this particular disease called cardiomyopathy. But in all other diseases listed here, selenium deficiency is always seen. So if you can somehow increase selenium, you may be able to treat many of these diseases, including neurodegenerative diseases. Thank you. So selenium has a very interesting story. Okay, in 1930s, selenium was thought to be extremely toxic, and so everyone was very afraid of selenium. And then in 1943, some research showed that selenium in high levels, of course, is carcinogen, so you should not take that. However, in 1957, we came to know selenium deficiency causes Pichon disease and Cachet-Beck disease, mostly in China and other parts. And then they found that you really need so much selenium to keep you healthy. So you can see, from highly toxic, it became an essential trace element. Okay? And then it also became anti-carcinogenic. From carcinogen in 1966, research showed that it is anti-carcinogenic. Later, in 2001, my lab at Penn State showed that selenium has anti-inflammatory activity. So this skeleton left is washed, pretty much. Now, now, it's a very important element. Okay. But to understand the mechanism of selenium, okay. So what is also very interesting is that selenium deficiency, that I told you, is also seen in case of Alzheimer's disease patients. Okay? If you look at patients who have Alzheimer's disease, particularly elderly population, you can see they have a selenium deficiency. That can be seen in the plasma, erythrocytes, and nails, and so on. And you can normalize it with the diet, and you can see selenium deficiency is seen. And there was a paper that was published where they did some modeling analysis based on the selenium that is available in the soil, because selenium comes from the soil, like other nutrients, accumulates in the plants, and when we eat plants, we get selenium. Now, based on the total amount of selenium throughout the world, these people from Switzerland actually did a modeling study and found out that this is how it looks now. Okay, there are many areas that have poor, very low amount of selenium. See this, India is also kind of in deficiency state. But in the year 2080 and 2099, okay, how will the world look in terms of selenium levels? There is going to be a lot of selenium deficiency because of increased mining and a lot of other reasons. You will see a lot of floods and so on. You will see global warming. All that leads to more selenium deficiency. So it's kind of we need to supplement our diet with selenium. Well, supplementing selenium is not very easy because we need to understand how selenium works. Why I say that is, this is how selenium gets incorporated. Okay? You all know translation, right? Everyone should know translation, right? Okay. Translation starts with the tRNA, correct? And then you will get the tRNA onto the ribosomes and you get your protein. Now, what is very interesting about this element is it actually works in the form of a 21st amino acid called selenocysteine. Have you heard of 21st amino acids? There are 20 amino acids. This is the 21st amino acid. So what is the genetic code? What is the codon for selenocysteine? If you see this, you all know this chart. Where do you see selenocysteine? There is none. So how does selenocysteine get incorporated into proteins? It actually uses a termination codon. What you read in your books is only half true. What I say is, you all know UGA is a stop codon. But I'm telling you, UGA is not a stop codon all the time. In some cases, UGA becomes a selenocysteine codon. That is because there is a specific lipid loop structure in the 3' end of the RNA that makes this UGA, one of the UGAs, to be selenocysteine codon, and then the next UGA it stops. So whenever you have multiple UGAs and you have this particular sequence, it actually incorporates selenium as selenocysteine. And this incorporation occurs on the tRNA itself. So it's very different. So there are a lot of enzymes that are present. And if you look at any kind of mutations, there are a lot of mutations in these. So no matter if somebody has any mutations in these pathways, if you give them enough selenium too, they may not make selenium proteins. It is useless. So unless and until we know what the genetic makeup is, or if there are any kind of mutations, you cannot just supplement people with selenium. You need to understand more and more. So if you look at what the different types of selenium proteins are, there are about 23 or 24, the list is incomplete. There are 24 selenium proteins, And all these red boxes you see are cellulose residues. Okay? So you can ask me, what happens if there is no selenium? What happens is, when there is no selenium, the tRNA stops here. The protein does not get fully translated. The RNA becomes unstable and it gets degraded, and that is called a process called nonsense mediated decay process. That nonsense mediated decay process will degrade all the RNA. You will not get any of these cellular proteins. So the question is, what do these cellular proteins do? These cellular proteins, there are 23 of them, and this was one of our old papers where we discovered some of them. And these cellular proteins have some unique functions. I'll tell you about a few. What they do is, in say for example, neurologic disorders, in the case of Parkinson's, Alzheimer's, Huntington's, all these, there are some cellular proteins that are extremely important. If you have less of the expression of particularly cell T, GPX4, now you can actually see an increase in some of those neurologic diseases. People with thyroid issues, a lot of people have thyroid issues. There is a cellular protein actually called DIO1, 2, and 3 that is responsible for converting your T4 to T3. T3, as you know, is triiodothyronine. That is the active form of the hormone, right? Now, to convert T4, the precursor to the active form, you need cellular proteins. So if you just take iodine, but not take selenium, you won't get it. You are just simply adding more iodine to your system. Okay? So then, in physiology too, it is very important. Thanks. How about in the brain? So in the brain, this is a heat map of looking at all these cellular proteins in different areas of the brain. Cerebral cortex, in the hippocampus, cerebellar cortex, and so on. You can see that there are some where you have very high cellular protein expression. The red one in high means it's very high expression. So some of those, there are about 5 cellular proteins, 5 or 6 cellular proteins that are extremely expressed in the brain, various regions of the brain. So if you have low selenium in the diet, your brain is going to be low in selenium as well. So you can ask me, what do these cellular proteins do? What these cellular proteins do is they help in reducing loss. How many of you have heard of loss? Reactive oxygen species. Reactive oxygen species come in various flavors. Hydrogen peroxide, then nitric oxide, then hydroxyl radical, superoxide. All these are considered reactive oxygen species. Okay. Now, where do these form? In all of you, right at this time, you are making a lot of loss in every cell in your body. But that is somehow suppressed by a lot of other enzymes. And so that keeps the loss under check. Okay? Because your mitochondria, your metabolism, everything always makes loss. Your mitochondrial respiration actually makes a lot of loss. However, the loss is kept under control by certain antioxidants like vitamin C and lipoic acid, selenium. All these antioxidants keeps the ROS levels balanced. If you have more ROS, what happens is it can be seen in every disease that you can think of. Okay? So some of these enzymes that are called antioxidant scavengers in cells are very important to keep your raw cell in check. That includes these enzymes listed here. I'm not going to repeat all of them. Just tell you about SOD, catalase, and glutathione peroxidase and thyroxine are some of the enzymes. Now, what they do is they act enzymatically and reduce the loss like hydrogen peroxide, superoxide, hydroxyl, all these they reduce. And that keeps the inflammation down. One of the enzymes is extremely very important in this case is an enzyme called GDX4. GDX4 is a glutathione peroxidase 4, which is also a selenium protein. What this does is, whenever there is inflammation, your membrane has a lot of phospholipids. Phospholipids can get easily oxidized. And when they get oxidized, they form what are called phospholipid hydroperoxides. These phospholipid hydroperoxides are extremely toxic to the cell because they are not only hydrophobic, they are also hydrophilic. So this anti-lactic nature of some of those phospholipid hydroperoxides can go into the DNA, into the nucleus, and cause a lot of mutations. And that is thought to be one of the reasons why some of us get cancer, some of them don't, and so on. So somehow we have to reduce this hydroperoxide back to its alcohol. What does that is GDX4. Now GDX4 is very interesting because if you look at the brain, dopamine is a very important neurotransmitter, right? Now, dopamine can get easily oxidized to this dopamine quinone or DAQ. Now, the dopamine DAQ metabolite can go and modify this GDX4 and kill it or inhibit the GDX4. So what happens is, if there is that oxidation because of loss of dopamine, that can kill GDX4, so now the stress increases even more. Okay? So there are quite a few neurotoxins that we know that impact the brain, and many of the selenium proteins are involved in this process. As a result, what happens is when we don't have selenium, and those selenium proteins are underexpressed or low, what happens is it leads to oxidative stress and loss of dopaminergic neurons, which actually then is one of the reasons why neurodegenerative diseases start and continue to happen. So you have to somehow change this process and see if you can reduce oxidative stress. One way of doing it is to give selenium and see if that happens. As I mentioned earlier, selenium proteins are the ones that mediate the effect. What happens is when you take selenium, liver synthesizes some of the selenium proteins, particularly one big selenium protein that goes to the neurons, and that then synthesizes all in the brain. It synthesizes all the selenium proteins and helps in keeping that low. However, when you have low selenium, what happens is you can see plaque formation increases, there is neurodegeneration, in Parkinson's, you see the vulnerability of dopaminergic cells actually increases. It also causes neurodegeneration. And so, it's a very clear association between low selenium and increased Parkinson's. So, what we decided to do was to see how can we understand more about the role of selenium in the microglial cells or macrophages in general. So we designed an experiment where we took a mouse that was, on no selenium diet at all, completely deficient in selenium. We took the femur, you all know where the femur is, correct? From the femur, we took the bone marrow progenitors and we cultured the bone marrow and made macrophages. So these are primary macrophages. Takes about 7 days. And then we treated them with an inflammatory stimulus called endotoxin. okay, LPS, like polysaccharide, for various times. And then we took the cells, extracted the protein, we also extracted some of the metabolites from the cells, and did two parallel assays. One is called proteomics, it's called high throughput shotgun proteomics. The other one is metabolomics. And what we were looking for is what are the different changes in selenium when you treat them with selenium or without selenium and inflame those macrophages in terms of protein, in terms of metabolites. Okay, these are the different selenium proteins I showed you. And our assay covered a lot of selenium proteins. These are the red ones are all selenium cysteine, and the blue area shown for each selenium protein is the peptide coverage when we did proteomics. Okay, so we pretty much covered a lot of protein. In each one, we had at least one or two sites for estimation. And we could do some good quantitative proteomics. And as I told you, we also did some metabolomics. So this is, I'm sorry, you cannot be able to read all the metabolites. And this is metabolomics. What metabolomics does is it's a mass spectrometry based assay where you can actually measure a lot of metabolites that you make in your cells and quantitate them. So this is color coded. Dark blue is low, dark red is high. Okay, and in between you have multiple different levels. And this is all very highly statistically evaluated and all that. And so what we saw was when you treat cells with selenium, macrophages without selenium and with selenium and increase the inflammation, you can see these cells group to different areas. This is called a test plot, a very highly statistically advanced way of graphing when you have multiple variables. In the testing plot, you can see there are multiple areas that are lighting up. It's beautiful to see. And what this means is that when cells without selenium and with selenium, or let's say you take one area, like 20 hours, you see that and this color are two different places, which means they are behaving differently under these conditions. And what the difference is, you can see lot of metabolites, the PCA cycle, glycolytic cycle, pentose phosphate pathway, all those change. Okay? Now you can ask me, what does this mean? Even in the case of enzymes too, like proteomics, we saw PCA is upregulated with selenium. Glycolysis is suppressed with selenium. So based on that, we came up with this very nice paradigm, and that is when you activate cells with a toxin, macrophages, without selenium, glycolysis pretty much takes up a lot of it. And you get a lot of lactate. But when you add selenium and selenium proteins, instead of stopping at the glycolytic pathway, it goes further and you get these other pathways, particularly pentose phosphate pathway and PCA cycle, and that generates more and more energy and it helps. This is very similar to what is called the Warburg effect. How many of you have heard of Warburg effect? Do you know who Warburg is? Have you heard of Otto Warburg or Warburg manometer? So, Dr. Warburg was a physician scientist in Germany, and he got his Nobel Prize in 1935. Okay? And what Warburg did was, he actually showed that you can accumulate lactate even under aerobic conditions. You don't need anaerobic conditions to accumulate lactate. And that typically happens in cancer cells. They're not in anaerobic state, but yet they don't go into PCA cycle and it stops at lactate. The normal cell will go into oxidative phosphorylation and you get less lactate. This, under aerobic conditions, accumulating lactate from glucose is called Warburg effect. He got the Nobel Prize in 1935. Okay. And one fun fact for some of you is I have some relation to Dr. Walker. And that is, my boss actually worked with Walker's one of his students. So that's where I'm related. Maybe that's why I like metabolism so much. Okay. But I don't think I'll get a Nobel Prize. This is the closest I can get to that. Okay. So when you change metabolism, what happens is selenium actually helps in the solution because it now increases energy output and helps reduce loss and helps in resolving inflammation in this particular model that I've shown you. I'm not going into the details of how we did all this, but what this does is it actually calculates something called the resolution index, and you can see selenium supplementation with the diet in these macrophages actually helps in resolving inflammation caused by herpes. So during inflammation, we see that a lot of these selenium proteins are expressed, and some of the selenium proteins are expressed at much higher levels, and that was something of interest, and this is based on mass spec analysis. So we found that without those selenium proteins, you have huge amount of inflammation, like TNF alpha goes up. TNF alpha is a liver necrosis factor. It goes up, skyrockets, and when you add selenium, it's suppressed. Same thing, there is an anti-inflammatory enzyme called arginase 1 that increases with selenium. So this is the other side of the coin. So based on this, we can say selenium downregulates inflammatory pathways and upregulates anti-inflammatory pathways. So what we did then was, we also showed that NF-kappa B is highly regulated by selenium. And that NF-kappa B is important because in TNF-alpha gene promoter, you have NF-kappa B binding sites. So doing something called a chip assay, we were actually able to show that that is because if selenium changes the escalation of the histones that are on the promoter of these TNF and COX2 and other inflammatory genes, and by changing some epigenetic mechanisms, it actually suppresses inflammation. So, inflammation resolution actually involves not just production of these different metabolites, but also a process called epigenosis, where when you have inflammation, you have all those cells that are causing inflammation. They need to be cleared off quickly. How do you clear them? You clear them by these macrophages. You teach the macrophages to clear the debris, and that debris, once cleared, actually helps in resolution. So somehow, even in the brain, if you can clear the debris, you can actually cause resolution. So we created some genetic knockout mice. So you take a normal mouse, and then you engineer certain sequences around the genes of interest, and then what you do is you cross those mice with a pre-recombinase such that you can delete genes. This is the old method. Now the new method is using CRISPR. You all heard of CRISPR, right? So using what is called a guide RNA, you can actually inject the guide RNA with Cas9 enzyme and that into the cycle, and then when you get the blastocyst, you actually delete that particular gene. And so that blastocyst can then go on, you can implant that into a pseudo-pregnant mouse, you can actually generate a knockout mouse. Now, this is a much easier way of doing knockout strains, and that's exactly what we do in the lab. We make a lot of genetically different mutants of mice. Okay. So we created one mouse where, you remember the tRNA that makes that selenocysteine? We knock the gene out for that tRNA. So that no matter how much selenium you do give them, they don't make any selenium proteins. Now, that mouse can be crossed with a 3-recombinase driver promoter of any particular gene that is of interest to you. For example, let me illustrate. Here, I show TRSP FOXFLOX lysine 3. What that means is, what I've done is that, sorry. What I've done is I have made all the macrophages in this mouse completely have selenium proteins. All other cells have selenium proteins. Only macrophages don't have selenium proteins. So it is called a cell-specific knockout. Okay? Now this technology actually enables me to now ask the question, what do these selenium proteins do in macrophages specifically? So we use this model to look at any kind of infections or whatnot, and we take these mice, put them on different diets, containing different amounts of selenium. One is deficient, one is adequate, one is supplemented, one is very high, and then carry out our experiments with whatever disease model. I'll show you one with colitis. You all know colitis. It is ulcerative colitis. It's a inflammatory bowel disease, and it's just inflammation of the gut. I know this is a neuro conference, but I don't know how many of you know there is a big connection between the gut and the brain. It's called the gut-brain axis. It's very important. Okay, so what we eat is really important for your brain. Okay, so your gut needs to be in good order all the time. So what happens is when we put these mice on different diets and inflame them using a chemical or dextrin sodium sulfate, you can see that the mice on isolating and supplemented sodium don't lose their weight. And if you look at the colons, the colon is very short in these two, but not in these two, which have isolating. If you look at the histochemistry or the intestinal pathology, this is colon sections, you can see completely loss of that structure. Here, this is beautiful villi. You see the villi, right? You can see the villi here. So it tells you that selenium supplementation actually helps. So we took this model and used the model where we have only left. We have used these knockouts, the specific macrophage knockouts. And that said, you give them high selenium, let's see what happens. If macrophages are important in colitis, even if they have high selenium in their body, macrophages lack selenium protein, can they show the same phenotype as selenium deficiency? In fact, it is. Here, these mice are very high selenium, but yet they have the disease. So what that tells you is macrophage selenium proteins are extremely important in reducing inflammation. Okay? We also looked at it in an intestinal infection model with a E. coli strain, and in E. coli too, we see the same effects. It's done by one of my former students. She showed that if you use those macrophage knockouts, they die. They can't take the inflammation when you give them E. coli infection. Simulating what happens if you eat bad food, you get stomach upset. That's exactly what it is. So, we'll move on. Okay, so there was one of my colleagues in the University of Bonn in Germany, and what Uli did was he made knockouts of this TRSP gene, but used a tree that deletes these in the microglial cells and neuronal cells, actually in neuronal cells, not microglia, neuronal. And what he showed was when you delete the cellular proteins in neuronal cells, you actually get a lot of damage. And that kind of is very similar to what happens in the Parkinson or Alzheimer's models. Okay? So cellular proteins seem to be very important. Last point is, I told you, efferocytosis is very important. Efferocytosis is nothing but eating dead cells. Gobbling them up. Okay? Now, when the cells are dying by either apoptosis or pyroptosis or ferroptosis or any of these different processes, these cells start displaying certain proteins. Those are called eat me signals. Okay? Yes, they are called eat me signals. Now, those eat me signals are recognized. They are membrane proteins. They are recognized by the cells, the phagocyte cell, and then they keep those up. So potentials keep these signals, and the proteins on the heteroside are called find me signals. Now those are under control of a lot of genes. So one of them is under the control of cellular proteins. And what we saw was when you have no cellular protein, for example, in this case, this group actually deleted seleno K. It's a seleno protein in the microglia, and they found that it actually has an Alzheimer's phenotype. So these are all brain sections showing exactly what is going on. These dying neurons are not taken up by the microglial cells because they cannot heterocytose or brain cells. Okay? We showed that this was recently published. We showed that in a mouse that lacks selenium W, these cells cannot phagocytose dying neutrophils. They are very low. And we did this by a method called flow cytometry based imaging microscopy. So this is combining confocal microscopy and flow cytometry together. So as one cell is coming through the flow cytometer, you can picture that, take a picture, and based on the fluorescence, you can see if the cell is being eaten up or not eaten. Okay, very simple technique, but it takes a lot of time. And we see that there is a difference. So, in summary, what I've shown you is that when there is inflammation, a lot of inflammatory mediators, including ROS and all that, increase. But if you increase cellular proteins, that inflammation comes down, and then there is a resolution phase that involves this heterocytosis, and that helps to reduce inflammation and increase resolution. That is probably a very important. With that, I would like to thank my lab members and a lot of collaborators at Penn State. This is my colleague, many external collaborators, funding sources, and all that. And I will leave you with this particular slide. This is my academic inspiration. Okay, slide. That is my brother, who retired recently as the secretary in the Department of University of Agriculture and Farmers Structure. He was a Chandra College product. Okay. This is my mentor, who was a promoter, a surgeon promoter in the Indian Navy. He fought in 2 wars, China and Pakistan wars. He was also decorated in the ASM. He's also a product of Chandra High School. My professors was a patient at the Department of Chemistry in Regional Institute of Education of RCE, and his wife Shubha. Because of their mentorship, I became interested in chemistry. I still love this habit. Professor Madhusudan, you probably all know Madhusudan. He was a small man. Another inspiration, my professors in the Department of Chemistry. My former PhD advisor, Dr. Ramdas, who was associated with one of Harvard's colleague, and my later postdoc mentor, Dr. Ray. All of them are my academic inspiration idols. So with that, I will take any questions. Thank you once again. Sorry if I have confused you or anything. I'll be happy to answer questions.
Now the session is open for discussion. If there is any doubts, please ask. Some questions.
Sorry, it's a very talk. You started your talk. I want to go for a general question. Most of the anti-inflammatory activity we used to study by taking phospholipid 2, not 1. What will be the reason?
Sorry, sir, can you say that again?
Phospholipid 2 we used to study, and in order to determine many chemical compounds in order to find out the activity of the chemical compounds. We used to study the anti-inflammatory activity or anti-tumor activity with respect to phospholipid 2. Phospholipids. Yeah, phospholipids. Like this. That is 2, not 1. What will be the reason? That is the general question.
I think phospholipids 2 would be more appropriate than phospholipids 1. Because phospholipids 1, even though, let's say, if you take out one of the esterified fatty acids, it will become a lysophospholipid. A lysophospholipid is important because it will activate many downstream pathways of, say, protein kinase C and so on. And as a result, what happens is it increases inflammation. So perhaps that is why you are looking at it. But another way of also doing it is doing a phospholipid 2 acid. That would be even more specific.
Another thing is selenium protein. This protein is main part we are studying.
I have studied selenium protein W, GPX 1, thyroxine reductase 1 are my favorite selenium proteins.
So Keshav is very close to me. I basically from PhD and Matsudan is a housemate to me.
I
I just came from the same institution here and we joined as a PG coordinator in PhD.
Oh,
And
Thank you for the wonderful presentation, sir. My question is that a few years ago I read in the newspaper an article about how Southeast Asian diet actually helps prevent and reduce neurodegenerative disorders like Parkinson's and Alzheimer's. So could it be that perhaps in Southeast Asian countries our diet has a higher amount of UC that they help?
good question. Yeah, South Asia, even for example, South Indian diet. And if I were to pick one particular food, I would say just your sambar. Okay, is actually a very highly anti-inflammatory food preparation. The reason being, sambar has turmeric. Turmeric has curcumin. Curcumin is an NF-kappa B inhibitor. Okay. In addition to that, you also have black pepper. Black pepper is used from long time before we even knew how black pepper works. Black pepper has a compound called piperine. Piperine actually helps in absorption, not just of curcumin but also other things. But when you have a lot of curcumin along with black pepper, which we use in making sambar, it actually helps absorb the bioavailability of curcumin increases. As a result, it actually gives you excellent anti-inflammatory effects. So, go sambar.
Is it possible to reduce the inflammation caused by nanoplastic nowadays through this cellular supplementation?
Yes, the question is, can we reduce inflammation by using nano selenium formulations? Yes, it is possible. In fact, plants actually make these nanoparticles with selenium. And if you can isolate those nanoparticles with selenium and you treat them, you can actually increase the absorption of selenium and it actually helps in reducing inflammation. Yes. Any other questions?
Thank you, audience, and I would like to welcome Dr. Devika, Principal, Shadowless College, and also I would like to call upon Dr. S.S. Malini, Chairperson, Department of Studies in Genetics and Genomics, University of Mysore, for facilitating our today's keynote person, Dr. Professor Sandeep. The audience to take a tea break and then we'll be back for the second session. Thank you. Yes, the audience to multiple back by 9:20.
ವಾಟ್ಸಪ್ ಯಾಕೆ ಕನೆಕ್ಟ್ ಆಗಿರೋದು ತೋರಿಸ್ತಾ ಆನಂದ್ ಸರ್ ನಂಬರ್ ಇದೆ ನೋಡಿ ಆನಂದ್ ಸರ್
ಪ್ಲೀಸ್ ಮಾಡ್ತೀರಾ?
వచ్చి ಹೇಳು ಶಾರದಾ ಕ್ಲಾಸಲ್ಲಿ ಲಾಟ್ರಿ ಇವತ್ತು ನನಗೆ ಒಂದು ಯಾವುದಕ್ಕೆ ಕೊಪ್ಪು ನಿಮಗೆ ಏನು ಮಾಡ್ತೀಯಾ ಏನು ಮಾಡ್ತೀವಿ ಕೊಪ್ಪು ಗೊತ್ತಲ್ಲ ಅದೇ ನನಗೆ ಮಸ್ತಲ್ಲ ಮಾಡ್ತೀಯ ಪೂರಿ ನೀನು ಯಾವಾಗಲೂ ಒಂದು ಹೇಳ್ತೀನಿ ಕೇಳು ಇಂಟರ್ನ್ಶಿಪ್ ಅವಶ್ಯಕತೆ ಇದೆ ಆದರೆ ಈಗ ಸಿಚುವೇಶನ್ ಹಂಗಿದೆ, ಆ ಸಿಚುವೇಶನ್ ಅವನು ಮಣ್ಣ ಹೇಳು ಇಬ್ಬರದು ತಪ್ಪಿದೆ ಅಂತ. ಮತ್ತೆ ಇನ್ನೂ ತಗೊಂಡಿಲ್ವ ಫೋನ್ನ? ಮಗಳೇ ಹೋಗು ಪ್ಲೀಸ್.